All right, let's get started. Can somebody confirm if you can hear my audio? Thank you, Hannah. So hello, everyone. My name is David Ruck. I'm here at the University of Maryland in Baltimore, Maryland, and welcome to Cytometry in R. And today we're going to be talking about applying transformations. So this is our seventh week of the course, and at this point we're going to take a look at how to apply transformations. So we need to be able to visualize both also the raw values of our flow cytometry data and then see how we are transforming them, i.e. how we're scaling them so that we're better able to resolve both the positive and the negative populations that are present within our typical flow cytometry and mass cytometry data. So there are several commonly used transformations when it comes to the data sets that we work with. For flow cytometry, oftentimes it will be either the logical or the bi-exponential transformation. If you're working with mass cytometry or cytoph data, more often than not, we see something more like the R-sign transformation. Regardless of what method of transformation you use, the goals remain basically the same. We're trying to rescale the data in a way that allows us to better interpret a positive from a negative population. If we end up misapplying a transformation, this can be dangerous for our analysis because that resolution is no longer there and we're not actually able to decipher our populations correctly. Therefore, what we are going to try to emphasize for today is that we always need to ensure that how we've transformed our data visually makes sense. We should not accept the default transformation for any function in R. We should tinker with it a little bit just to make sure that in the context of an exploratory data analysis, that it all is working as we expect. Additionally, we have near perfect timing because the Cytobytes YouTube channel, which is one of the ISAC-led Education Committee initiatives, recently put out a short video about why do we transform our data, and Felix did a very good job, so I'm linking that article here if you want a different way of viewing this particular aspect. So as we do every week, I've updated the Cytometry in R course repository with all the updated data sets and code. So if you haven't yet done so, please go ahead and sync your fork and then bring in the code locally to your local computer using a pull. Once it's all available locally, just copy the data from the Cytometry in R course repository folder over to your own weekly project repository. If you're following the course organization structure, you'd likely name it something like week 7. And then after you've set up a few subfolders and have everything set up properly, just make sure you're committing it to version control and pushing it to Git. That way, if worst case scenario something happens to your computer, you're not losing out on all your hard work. All right. So this week, we're going to be extensively using the Flow Workspace package. Basically, picking up where we left off a couple weeks ago during week 5 when we were building, loading in our FCS files into a gating set object which we could work with. So when we did this back in week 5, we were loading in a fully assembled gating set via the CytoML package, basically being able to pull in a FlowJo workspace. This time around, we're building our own gating sets from scratch, so we're continuing where we left off that week, but just seeing how we go about transforming our own data. Consequently, because we need to make sure those transformations are applied correctly, we're also going to be using the ggcyto package pretty extensively throughout the day. So because we're going to be using these packages quite a bit, and we're going to be using various different functions that are found within both of these packages, it makes sense to go ahead and attach them to our local environment using the library call, so that's what we should go ahead and do. So we go ahead and call both library for Flow Workspace as well as ggcyto. And then the data sets that we'll be working with today are going to be 2. We're continuing using those SpectraFlow cytometry data set we provided a couple weeks ago, namely those 6 unmixed SpectraFlow cytometry files first shared back during week 5. This time around, rather than loading them in via the FlowJo workspace, via the CytoML R package, we're just going to assemble them into their own gating set via the Flow Workspace package. So the one thing to remember for these particular 6 SpectraFlow cytometry files is they all share 2025 as part of their file name. So we're going to use that part of their shared common names to isolate them out when it comes to list.files argument. Vice versa, we're also going to be working with a mass cytometry dataset. So for those of you who do mass cytometry analysis and who have been patiently waiting for the last 6 weeks as we've been going through a lot of spectral flow cytometry data, I'm bringing in some datasets for you. The tree files in question came from the import repository, specifically study SDY2739, which was a clinical immunity to malaria study. So I borrowed three of their files. They all start with G1_2 as part of their file name. So when we're setting up our list of files, we'll go ahead and isolate them to their own gating set. The reason for this is Flow Workspace notices when files are being loaded into a gating set that don't have matching number of columns or have different fluorophores or metals as part of the column names. So we just need to make sure that each of the data sets is kept in its own separate gating set. So how we go about doing this is kind of similar to what we've done so far in the course. If you're keeping the organizational structure for your setting up your local project folder, all those FCS files will be within the data file. So we just set up this initial file path argument pointing to data, and we're saving this to the object variable storage location. And then we are having the list.files function search within that location, look for anything that matches the pattern.fcs, and then because we want to have the full file pass between our working directory and those individual FCS files, we're going to set the full.names argument equal to true. Once this is done, we're going to have just the general list of files for FCS files. Now, remembering that there's shared parts of the names that will distinguish between the spectral flow cytometry files and the mass cytometry files, we're going to go ahead and isolate them out from this broader overall list. The way we're doing that is kind of like what we saw a couple weeks ago. We're using the little square bracket notation and then the stringer packages structure detect function, which is basically going to look through each of those individual file paths, and anytime it encounters something that's equal to 2025 within the file name, it's going to come back as true, which in the presence of that square bracket is going to become an index value that then gets isolated out, basically being able to select out the SFC files by themselves. And vice versa for the mass cytometry dataset, where they all have the G1_2 in part of their file name, it's going to return for true for those particular file paths, isolate them out from the rest of the spectral flow cytometry data, and that's how we're going to isolate out our cytoph files. So let's first go ahead and start tackling our SFC dataset. So one thing we should always go ahead and double check is just make sure that our setup to isolate out the particular file paths did indeed work. So let's just go ahead and print out SFC_files, and sure enough, we can see the isolation of the patterns that contain 2025 did indeed correctly work, and we have the same data set we've been working with for the last couple of weeks, namely cord blood specimens that were either controls or treated with SEB. Circling back to what we had done a couple weeks ago with the week 5 lesson, we're going to load these in to our local R environment using the load cytoset from FCS function. That way we're not using up too much active RAM memory on our computer. And just like we did that time, we're using load cytoset from FCS function. We're providing it the list of file paths, and then we're setting the truncate max range and transform arguments equal to false, kind of like what we've been doing throughout the course. This now becomes our cytoset object, which we then pass to the gating set function to go ahead and create our gating set, which is that operational unit that we're working with within R. And as you can see, for our SFC gating set, it's coming back and showing that it has the 6 files present within. So one worthwhile aspect that we should always go ahead and do with our gating sets is just to check to see what the metadata looks like at the current time. So as you may remember from a couple weeks ago, the way we do this is using the pdata function, which we can just use place our gating set's name in the middle of the little parentheses, and then go ahead and check. And sure enough, you can see that we have the 6 files that we're interested in, but also for all we have is just a single column called name. This is just the row names on the side, so we don't have much metadata with which we can actually go ahead and sort these individual specimens. So, however, if we go back and look at the existing names of these individual files, you can see there's some little bits of metadata already present. We can see the date of acquisition for these files is kind of like towards the beginning. We can see the experiment series number for alpha beta number 2, meaning it was the second alpha beta experiment. You can see the specimen ID right there with the infant. You can see the control or treatment condition right there at the end, right before the .fcs. So we do have additional useful information, but we don't have it yet within our particular metadata for this gating set. So it'd be useful if we were able to extract out that information from the file name and just kind of create it as its own separate columns in metadata so that we could then actually use it for a filter condition to isolate out like just the SEB treated condition or just the control treated wells if we were trying to do so. So if we're thinking about how to do this, remember that our gating set is a complex S4 style object from Bioconductor. It's not going to allow us to directly manipulate it as far as what we're able to do. So if we want to be able to use like tidyverse functions to kind of clean up the file path name, let's go ahead and assign it out to its own object variable. So basically that same line of code we just saw, where we're just checking the metadata, we're assigning it to the object variable metadata. And when we're just checking the str function to see what's happening behind the curtains, we can see metadata is just basically another data frame that we've encountered throughout the course, more recently within the tidyverse week 4 class. And this is one that we can use the dplyr package to kind of clean up that particular file name, make additional columns, and then be able to do a lot of the data manipulation, data tidying process we've seen for the last couple of weeks. So let's see what we can do when we are actually working with the package dplyr. So let's go ahead and load in dplyr so we have access to all those data tidying functions. And because we are likely to do stuff similar to what we do with structure detect when it comes to extracting out particular patterns, let's also go ahead and load in the tidyverse package stringer by calling the library to get it attached to our local environment. Now, we don't want to accidentally mess something up and overwrite the existing name information for that particular column. So especially when you're doing data tidying, it's always good just to kind of duplicate an existing column and then tidy on that new column so that worst case scenario, everything remains intact. So the way you basically duplicate a column in R using the tidyverse is basically we're using that mutate function. So we're passing our data frame through the pipe to mutate. And then basically that particular column name, which is set as name, we're assigning it to a new condition column, which is going to basically come back and show us we now have 2 columns present. So if we glance at the current names of our FCS files, we may notice towards the end, right next to .fcs, is the particular condition treatment. Namely, are they control treated? Were they PPD well treated? Were they SEB treated? And this is between the very last underscore and the very first .fcs. So if we're thinking about how do we extract information that we have present within these individual file names so that we're just left with control or we're just left with PPD, we're basically trying to find a way in R to get rid of all this extra part of the character string right before the particular condition and then get rid of the last part, i.e. the .fcs portion. So we're going to need to figure out a couple ways in R of how to clean up these file names that are within this condition column we just duplicated, just so that we're left with just control or just SCB respectively. So within base R, there are 2 functions that can do this, namely the sub function and the gsub function. These rely on providing a regular expression, also shorthand as regex pattern, which they then match, and then you can provide a replacement value that they can be swapped in its place. However, if you don't provide a replacement value, if you just leave it as quotation marks by themselves, it will just replace it in place, not replace it with anything, and therefore you're able to get rid of extra information. So consequently, if we're going to try to use this base R subfunction, we want to have something that will basically replace everything up through the last underscore, but just leave it empty, which would just leave us with control.fcs or seb.fcs as the particular file name at that stage. So the regex expression that we're going to see used at this first step here is going to be this one, basically .*_. We'll explain how regex works in a minute. I'm just showing you how it looks like in function, where basically it's getting rid of anything that's present in front of the very last underscore, and it's replacing it with nothing. As you can see, there's the 2 quotation marks next to each other, meaning nothing being given. And it's acting on our testing data frame, and this little $ accessor is basically selecting just the condition column. So you can see when we run this particular line of code, we clean up all the first part of the file paths. It's no longer there. We're just left with control.fcs, seb.fcs, etc., etc. Now, we could continue using this modification that we already did, and then just target the .fcs portion, and also just end up leaving the replacement value empty as well. So for example, if we just ran that first line of code, we had left control.fcs, seb.scs, etc., etc. We're just passing it again to the sub function. As you can see, we're providing a new pattern, .fcs, the replacement value is being left empty, and then it just ends up deleting that last portion, .fcs, leaving us with just the values that we're actually interested in. So I had mentioned that the regular expression pattern was asterisk underscore. So please be advised, even after years of practice on my end, I will often still struggle when it comes to remembering what's the proper regex syntax code to be able to extract out a particular pattern, especially when it's like, is there a difference between the second and the third underscore, the final underscore, the first underscore? Regex is kind of complicated, especially as you come from a biology setting, first learning how to do more the computer programming sense. So this is not an area that I recommend like investing a huge amount of time in. One thing I found particularly useful in the last couple of years is that if you provide a large language model, kind of like ChatGPT or Claude, a very narrowly focused question, it can give you back the answer as far as what the relevant regex code is relatively quickly. So for example, for this particular problem, if you're asking your favorite LLM, you need to specify I'm working in R, so it doesn't return you Python code or something like that. Using a particular function, using sub, because there's multiple ways of doing things in R. And basically you want to keep everything in the file name after the last underscore. It will more often than not give you the proper regex code in the application with the sub function to be able to do so. So in addition to both the sub and the gsub functions in base R, we can also get a very similar outcome with the stringer package from tidyverse, namely using the structure extract function. So it also has regex inspired hieroglyphics that can be kind of convoluted when you're trying to do them by hand. So the main things to note for the next line of code that I'm going to show you is you're not expected to know exactly how it works, but a few guiding principles is if you ever see the upward pointing arrow, it's basically denotes the very start of a character string. And then the $sign basically serves to indicate the end of a character string. So basically when you're trying to extract out a particular pattern from a file name, you're basically providing the bits and cues pieces basically to focus it in on what it needs to focus. So in this case, if we wanted to do the exact same thing we did with two subfunctions, we're using the structure underscore extract function, and we're passing in the testing, specifically accessing the condition column. And basically that first argument between the square brackets, it says from the beginning go to the underscore, and that's what's being gotten rid of for the first part. Then you're keeping all that little bit intermediate until you hit the section that's .fcs, where that's the very end of the character string. And as you can see, this convoluted hieroglyphic of regular expression ends up extracting out the variables that we're actually interested in. So wrapping this particular regex line of code, we could then use it to extract out a particular pattern from a file name of interest. So how would this look like in practice? Well, remembering our pipes, we're basically taking our data frame, metadata, passing it through the pipe to the mutate function, which is basically creating that new column condition, which is identical to the current named column. This little intermediate gets passed through the next pipe, once again to a new mutate function. We're basically writing back on to the condition column, where we're extracting from the current values everything that's between the last underscore and the .fcs. And as you can see, we just get returned back a new condition column that's been cleaned up, showing us just control just SCB. Fortunately, when it comes to extracting out a pattern, it's much easier than trying to grab something that's not the beginning and not the end. So if we want to create a specimen column based on those ID numbers, you can see we're just adding on another pipe. But instead of this convoluted mess of regex-inspired hieroglyphics when it comes to the code, it's something a little bit simpler. Basically, it's matching a pattern I and F, and then any 3 additional digits is what's being extracted out. So in this case, this is being assigned to a specimen column, and then we're just selecting the condition in the specimen column, and you can see this is the new additional bits of metadata that we could add in to our existing. So to actually update our metadata, we need to go ahead and we have 2 data frames. So we need to combine these 2 data frames together, basically adding in the columns from one into the columns of the other. So the way we could do this in base R is using the cbind function. And what this looks like is you have your first data frame, which is metadata, which just consists of the name, and we're combining it with our 2 extra columns data frame we just created in the last scene. And as you can see, we still have the row names, but now we have the 3 total columns that we're interested in for our actual gating set. And now to actually add them to our gating set, we just have to overwrite the existing one. So basically, if you see, we have this data frame, updated metadata, and we're just assigning it using the pdata sfc underscore gating set, which will overwrite the existing metadata, and now it's updated within our gating set. So this is kind of the process of how you would go about adding additional metadata to your particular gating set object. Now, the reason this can be useful is you can use it to subset your gating set later on in the future. So in this case, we only have like 6 FCS files, but let's say you had 120 FCS files based on different conditions, different treatments, different male or females that you wanted to subset. Well, the way you could subset a gating set in R is basically you provide your gating set, and then you specify the particular condition in the metadata, and basically using that percentage in percentage operator we saw from filter in the tidyverse, we're basically grabbing only things that have the SEB value. And as you can see, we subset it down from those 6 FCS files to just those 3 FCS files, just corresponding to those SEB. So this is one of the values of being able to actually manipulate the metadata for your gating set. All right. Now that we've gone down this detour of metadata and been a little tormented by regular expression patterns, we can go back and start looking at our FCC data set. So as we look at it right now, we can see that yes, it does have 6 samples. It's good to know what fluorophorus your particular panel may have, because oftentimes we have different versions of different panels. Or if you are working with somebody else's data or data that we grab from a repository, it may look a little bit different from what you need to do. So one way that we can go ahead and check to see what the particular fluorophorus for a flow cytometry data is using the column names function that we've already encountered. Because remember, within the expers matrix data frame, it still has each fluorophore corresponding to a column. So what this will look like if we run column names on our gating set, you'll see it returns back the individual fluorophores or other parameters that are present for the individual FCS files. So as we look at this, we can see this was, I think, a 32 color spectral flow cytometry file. It also includes side scatter, forward scatter, side scatter blue parameters, both for area, height, and width. And there should be a time parameter as well. So this is just typical what you'll see for many flow cytometry files. Additionally, we may be interested in seeing what the actual marker names corresponding to those individual fluorophores are. So we had encountered a way of doing this a couple weeks ago when we went actually breaking into the S4 object to basically find that parameter data, but that can be quite convoluted to do. So we'll just take advantage of the hard work of the developers and use one of their helper functions, namely the marker names function, just to retrieve out that individual piece of data that we're interested in. So once again, if you see we run marker names on a particular gating set, we get back those named lists that we've encountered in the past, where basically the name of the list corresponds to the name of the individual fluorophore, and then the value that's present corresponds to our individual marker. So this way we can see the actual markers that are present for a given FCS file. So at this point, we've looked at the column names and we've looked at the markers, and we're going to do a lot of plotting today. So let's go ahead and run the ggcyto package just to make sure we're able to plot the data correctly, that we all have the correct version of both ggplot2 and ggcyto installed on our own local instruments, local computers. And sure enough, this is the code we encountered back in the introduction to ggplot lesson. We can see that running ggcyto on just the forward scatter and side scatter, we get a kind of representative flow plot for what we expect. So please note, when we're actually using ggcyto operating on a gating set, we're just using the square brackets to index the first specimen present within the gating set. And when we're basically just using a single square bracket, we're basically subsetting down just to that individual specimen. So if we use the SCR function, just check to see what's happening to the class in the background, you can see it's still returning an actual gating set, similar to what we saw for the more complicated one with the 6 FCS files. However, if you had accidentally included 2 square brackets, it would actually have broken into the S4 object, and you'd be getting back something of a structured gating hierarchy, which kind of corresponds to the individual gates. So as you work with the individual arguments for ggcyto or for flow workspace, just remember the type of object you're passing to the function does matter, so keep an eye on how many indexes are being used. So let's go ahead and check something besides forward scatter and side scatter, because forward scatter and side scatter for flow cytometry data are very linear in form, so they don't often need to be transformed. Let's look at something that's untransformed data. So let's go ahead and check CD4 and CD8 since these are mostly T cells. So as you can see, that initial first plot we get back using just the untransformed data, it's very, very stretched for the CD4 and it's very, very narrow along the y-axis. Similar story for CD8, we can see the cells as far as the stretch, but it's still very, very not well resolved. And it doesn't really look like the flow cytometry data that we're used to working with. So this is typical for many fluorophores because although a lot of the space is being treated linearly, especially around that negative population, when it comes to cells that are staining for a particular fluorophore, there can be lots and lots of logs of difference between the cells that are just dimly staining for it, as well as those that are super bright for it. So if we're actually thinking about it, it's like there will always be like multiple logs out of events that would basically really, really expand out this x-axis. So part of the reason we end up transforming is we're compressing this more logarithmic space of the scale down to something that's actually manageable, either using like the bi-exponential or a logical transformation, while still keeping this region of the scale that's more behaving linearly as a linear fashion. So you may remember when we brought in the same exact file using the CytoML package originating from a Flowjo workspace, we didn't have this issue because it looks something much more similar to this. The reason for this is when we brought in the Flowjo workspace, all the actual transformation, compensation, etc. data was also brought in and incorporated already into the gating set. So since we're building our own gating sets from scratch, we need to provide this information so that we're actually able to visualize our underlying data in the same manner. So currently we don't have any transformations applied. The way in Flow Workspace that we could check to see if a transformation has been applied and what particular parameters, arguments, values we have provided to that transformation is the gh underscore get underscore transformations function. So if we run it right now, we can see it returns back as no particular value being present. It just says it's basically a list, but there's no values being provided. So the way Flow Workspace lets you the way Flow Workspace allows you to do transformations is it provides several default transforms. You can include logical, bi-exponential, R-sine, and a couple of others. It will also give you the option of creating your own from scratch, providing your own math formula, but that's a little bit outside the focus of this course. The main context of when you're working with it is this returns something that's known as a transformer object, which is basically a list that contains all the different arguments to both be able to transform your raw data into transformed data, but then also be able to reverse that process, basically an inverse transformation, to get back to the original appearance of your data. So for example, if we're using some of the defaults, there's the logical underscore trans function, which uses the logical transform. And if we're assigning this out and then using str to check behind the curtain, you can see the structure of this transformer style object. Namely, it gives you the name, it gives you the function for both the transform and the inverse. It tells you whether it's been applied yet, yes or no, and then also will tell you like where it expects to see breaks, etc. So some more of the nitty-gritty details of what's going on behind the scene. But the same process is happening for all the other transformations that we can apply to our dating set. So for example, if we're looking at the more bi-exponential transformation, you can see it actually has like the individual argument values, channel range, max value, negative and positive decades, as well as the width basis. It includes some default values, that is just what's present by default, which we can also go ahead and change. And then you can see there's also the inverse argument, which is used to convert back to the original version. And same thing for those who do mass cytometry who prefer to use the A-sign transformation. It's a similar style list, so this is what we mean by a transformer style object. And there's actually another A-sign transformation, which we'll see as we go forward. So as always, when you're working with a new function in R, it's always worthwhile to try to go back and see what's present within the help documentation, just so we're better able to see what the various arguments are and how to use them. So in addition to having the individual transformation that we want to use, we want to be able to specify what particular fluorophores or what particular metals we're trying to transform. Because oftentimes you'll have time or you have like forward scatter, side scatter, which is already behaving linearly. You don't want to end up transforming these by accident just because you passed all the column names. So if you remember, when we ran column names a little bit earlier for the spectral flow cytometry dataset, we got these particular values. So we want to be able to transform our particular fluorophores, but maybe we don't want to transform the forward scatter, side scatter, or time parameters, or maybe we want to apply different transformations to different particular fluorophores. So the way we basically need to select out which of these particular column names, i.e. which fluorophores need to be transformed, is we can use some steps that we've already encountered in the past. Namely, we can use the index method using those square brackets, and then combine a combination of structure detect and a negation symbol to basically exclude out individual column names that we don't want to include. So, for example, taking our column names for the SFC parameters, we can pass them in and using string or structure detect, find anything that's equal to either forward scatter, side scatter, or time. These little pipe symbols basically denoting or in the context of this particular argument of code. And then because we're including the little exclamation part, anytime this value comes back as true, meaning if we come back to the time value, it will return as true, but the exclamation sign will negate it, turning it into a false value, which means it won't be kept in the final vector. So this is how you can see we can basically quickly be able to remove out the forward scatter, the side scatter, the time parameter, just be able to exclude them out from our particular column names that we want to work on. So now that we have the particular fluorophores that we want to apply transformations for, and the particular transform that we want to apply, we just have to combine these two together into what's known as a transformer list. And this is done via the transformer list function. So the way this would be structured is we're using the transformer list function, and we're providing those list of fluorophores that we want to transform, and then that particular transform argument we've set up, in this case using the bi exponential. And then basically this will go ahead and transform it, and as you can see, it comes back as a list, listing each of the individual fluorophores, and then providing the actual transform being applied. If we went to behind the scenes and checked with str, you would see it's actually a list of lists where you have this individual item being the particular fluorophore that you're interested in, and then the individual transformer arguments that you've provided being applied. So this is a way basically how R knows, like, it doesn't encounter forward scatter or side scatter or time, so it doesn't end up applying them. And if you're trying to customize your list to apply certain arguments to one marker but not to the other, it would be happening at this level. And same thing, this is another example, but just using the APCA. As far as like we can see, these are the default parameters being applied at this point in time. So the very last step, now that we've created a transformer list that has the information of which fluorophores and which transforms are to be applied, we actually need to go ahead and apply it to our gating set object. And the way this is happening is we're using the transform argument, we're providing our gating set, and then we're providing the actual transforms happening. And as you remember, the gating set is working off of a pointer system, pointing at the storage location in memory. So we're not actually having to assign it to a new object, it's just working in the background at that point. Now, once we've applied the transformation, we can go ahead and replot just to see how it has changed the values, and we're going to get back something that looks like this. So right off the bat, we can see that those default arguments that were provided by the bi-exponential transformation are not quite right. We know this should look like a CD4 by a CD8 population of T cells. This looks like it's not really well compressing the negative region. Everything looks kind of artificially double positive in look. So this is not a good, this is a situation why we don't always trust the default. We should always tinker, we should always address them individually as we go. So how do we go about changing these individual arguments? Well, first off, let's go back and check the transformer function. So remembering when we're trying to access the help documentation, if you're within positron, you can just hover over the code chunk over that particular function name and be able to see it. Otherwise, just add the little question mark in front of it and hit run. And we'll get back to the documentation page that looks something like this. Now, this documentation page is not particularly helpful. It gives you only two arguments, ... which is basically parameters being passed to a different function, and then n, which corresponds to the number of breaks. So basically what's happening here right now is this is what's known as a wrapper function, where you're providing individual arguments to it, and then it's passing them to a similar function behind the scenes to actually do all the actual work at figuring out how to apply the transformation. So in this case, we need to actually go and check out what this FlowJoTrans function is doing in order to figure out the arguments that we need to change. So sure enough, if we check up the FlowJoTrans function, we can see that it actually comes back with a little bit more useful information. Under usage, whenever you see these particular values, these are the default values that are specified for that function, which is what we just ran. And then you can see the particular arguments being listed and what they do. All right, so let's go ahead and evaluate what these actual arguments do in the context of visualizing out our FCS files. So if we look at our current visual, this is kind of like what we're seeing on this plot. Now, because of how a gating set works and how it's basically pointing at a particular object somewhere on our actual hard drive or solid state drive, it's not such an easy thing to actually overwrite in place, especially once you've transformed the argument something like this. So for all these examples, you'll basically see me rerun the line of code creating the FCC, cytoset, and then gating set. The reason because of this is once you start pointing at it and then you drop it and recreate it, those pointers get refreshed, so there's no actual transformation applied, so you're able to refresh as you go. So in this case, basically you're loading in our files to a cytoset, creating a gating set, and then we're basically working with this initial FlowJo by exponential transformation argument, and these are the default arguments that are currently being provided. These are the ones that we're going to change around. So this is being assigned right now to by exponential, and then we're combining that by exponential with the actual fluorophores that we're trying to transform to create that transformer list, and then we're just reapplying it to the gating set. So currently using those defaults, we see it doesn't quite look right, so let's go ahead and figure out which of the arguments is actually causing the plot to look like this. And because we need to make sure it's actually overwriting correctly, we're going to go back sporadically just to check the get transformations function, just to make sure those argument values have indeed changed in the background, and that it's not just returning the same plot over and over again. So right now, if you check, now that we've applied those initial set of transformations, this is the value we get back, and you can basically see the individual arguments we provided for the transformation are present in the background. So before we go about messing all the other arguments, it's best to tackle width, because that more often than not tends to be what's messed up by the default in the context of flow cytometry data. So normally in FlowJo, most of my plots for my spectral flow cytometry data tend to be with a width argument of either -500 or -1000. So let's go ahead and start off with a -1000 value just to see what happens. So once again, just running the same lines of codes, recreating that gating set, applying the transformation. In this case, I've changed the defaults just to have width basis set to -1000. And right off the bat, my plot looks a lot more like what I would expect for my flow cytometry data. I can see my CD4s, they're a little bit stretched out on the positive, but then I see the CD8s nice and symmetrical right around the negative values across the board. So this looks much better, especially compared to what we're looking at. So this was at -1000. Let's just change around the width value a little bit more as we're testing. So once again, rerunning our lines of code. In this case, we're dropping it down to the -500 for the width basis. And sure enough, you can see the CD4 has shrunk a little bit. And because there's less compression happening within that negative portion, the CD8 has spread out a little bit, but it's still very much discernible as where the negative starts and where the negative ends. Vice versa, if we continue tinkering and we drop the width basis down to -100, rerun those lines of code, you can see the CD4 looks very much more constricted, but at this point we've lost any actual resolution of compressing the negative portion of the plot. Everything's very much spread out, so this is one of the situations we've gone a little bit too far. So if we're actually setting our width parameter, you could argue whether we want to use the -500 or -1000 to give a little bit better, more in line of what we expect to see for our data set. So now that we have width set, and for the next couple examples, I'm just keeping it at -500 so we can see the effect of the other arguments. Let's check out the next couple. So channel range is something that's normally specified by the instrument. It's currently set for 496. Just to double check to see what's happening, let's go ahead and double it. So if you look to see, we are keeping width basis at -500, but we've doubled the channel range default argument. And when we're actually going and plotting, it looks kind of similar to where we had left off. So no immediate apparent change. Vice versa, if we go back and check to see what happens if we have reduced it, we get back eventually the plot, and it still looks pretty similar. So at least channel range does not seem to be quite responsive in the context of our particular data set. It seems to be roughly in the range of where it needs to be. And vice versa, we should just go ahead and check to make sure those transformations were actually applied correctly. And as you can see under the channel range argument, it was indeed half the original value. So, relatively minor changes, no major obvious visual difference. Let's just leave it as it was for the default and just move on to the next argument. So max value is one that you'll often encounter when you're working with individual instruments. So I know like some of your individual instruments will show you the max value that's being shown. It will be something like around your 10 to the 5th or 10 to the 6th with a couple tick mark values, and that will correspond to a particular max value. So that's basically what's being represented right now in R. So if you have access to your instrument, you can look it up. If you don't have access to your instrument, like you're working with repository FCS files, you may need to adjust back and forth accordingly. So the default currently is set for 262.144. So keeping with that negative 500, let's go ahead and double this max value, just see what happens. And you can see instead of that little nice rounded CD4 population, when we doubled the max value, all of a sudden it's a little bit more stretched out. This makes sense because we're saying to R that is actually brighter values, so that's affecting the scale. So we basically phenocopied a change in the width basis that we've already encountered. Vice versa, what happens if we end up reducing the max value? So instead of doubling it, let's say we reduce it by half. And we rerun that line of code, and sure enough, we can see we start phenocopying something that we started seeing with width being set to negative 1000. So at least in context of how we are doing our transformations, we can see that the width basis and the max value are kind of interrelated. So what we set for one may affect the other. So this is good to keep in mind as you're tinkering with these individually. Additionally, some instruments will allow you to set different positive decades. Many are set at like 4.5, 5, 5.5, or even up to 6. So if you know what your instrument settings are, you can apply those directly. In this case, for Flow Workspace, the default is set at 4.5. Just visualizing what our current plot looks like, this is what we have. But let's say we go ahead and increase it a little bit. Let's say we can walk it up to 5, so an increase of half a decimal. And you can see, sure enough, we end up kind of phenocopying what we had for that width basis change. So max value, width, and the positive decades are all kind of interrelated, so just be a little cautious when you're doing your transformation. All right, and double check here. Similarly, especially if you're working with spectral flow cytometry data, often you'll have mixing errors that can result in very negative values. So sometimes you want to kind of like compress the negatives just so you're able to see them on the same screen. So they have the option within R of adding extra negative decades. The default is set at 0, so just reminding you this is what our current plot is set at. We can increase this individually, so let's go ahead and add it up so that we're showing an extra negative decade. As you can see, the plot has changed a little bit. There's a little bit extra spread on the negative population, but not very much. It's still actually resolvable, it's still discernible, so no major effect. By contrast, if we had doubled that to 2, and then visualize the plot, you can see it starts spreading out a little bit more. So you do have the option, but at least when it comes to these pretty relatively well-behaved floors, it may not be the best thing to tinker with. So in recap for the spectral flow cytometry dataset, we've looked at the various arguments and how they can affect the overall visualization of our data, and we've seen how some of these are particularly interconnected with each other. So it's oftentimes best to be able to verify what your instrument settings are, and then just use those as you're setting up your transformation in R. But if you don't have those, just go ahead and mess around with the actual individual values until you get good resolution, just remembering that they can be interconnected, so just be cautious on what you end up doing. All right, now that we've looked at the main bi-exponential transformations for spectral flow cytometry data, let's look at our mass cytometry, our CyTOF data set, where we more often than not will use the R-sign transformation. And let's just go ahead and see to make sure that we do have the correct files. And sure enough, they did isolate out based on the G1_2, a part of the name. So these are our mass cytometry files we found from import. And let's go ahead and load them into their own gating set object. So similar code as what we ran for SSC, being loaded in to a MC_gating set object. And at that point, let's just repeat our process just to see how many metals are present, what the individual markers that are present for them. So if we run column names on our MC gating set, we can see all the different metal isotopes get returned. However, if we are running the marker names just to see what's being present, you can see that this name list gets returned, and we can see that some of these patterns correspond to beads, some of them correspond to barcodes, some of them correspond to epitope tags. So we may not want to be transforming all of these. I will default to those who have more experience with mass cytometry as far as the actual pre-processing of what's going on. But in terms of just the general visualization for today, we can focus in on CD4 and CD8, knowing we haven't carried out all the proper cleanup steps when we're working with the entire FCS file. So if we visualize the CD4 and the CD8 population of this mass cytometry file, you see it's a common similar story. There's a lot of values right around 0, which is kind of typical for mass cytometry files, which is why oftentimes the R sign transformation is preferred. And then there's a long stretch of values that are positive, not quite up to the same log as what we'd see for spectral flow cytometry files, but still quite decent of a stretch. So, just like we did for spectral flow cytometry, we need to specify the transformation, but we also need to specify the markers that are being transformed. So in this case, let's say we didn't want to apply the transformation to those barcode or those beads corresponding metal isotopes. So we will actually need to separate those out from our particular markers of interest. In this case, the way to do it is basically we're just using the marker names on our particular gating set, which is returning out this named list. And then basically for that named list that we're getting back, we're just basically excluding anything that's coming back as a value of barcode or beads. So we just have our mass cytometry markers of interest at that point. Now, we can't actually pass the marker name when it comes to applying a transformation. We actually need to be applying the column name equivalent, in this case, the metal isotope. However, if you look at this one in its current state, you can see this is a named list. So the values that you get back if, for example, you pulled from this item would be the individual epitope tags. But if you're able to retrieve the names of the list, you'd actually be getting back the metal isotopes that we actually need for the transformation. So that's what we're actually going to be able to do. So if we use the names function on a named list, it will actually retrieve the names that are there, not the actual values. And this is how we get back the corresponding metal isotopes for the markers that we actually want to transform. And once this is done, the process is very similar to what we had for the SFC files. We're going to go ahead and create a transformer list that we can then check. So in this case, we're creating for the A-sign transformation, we're just using the default. We're combining it with our metal markers that we're actually interested in into a transformer list. As you can see, it looks very similar to what we had for when we used the bi-exponential scale. There's both a transform and an inverse transform accordingly. And then we're just actually transforming our gating set, which is then, if we visualize, you can see it looks a little bit closer to what it needs. There's still some adjustments that can be done back and forth, and we'll see those as we encounter them. So if you're actually interested in what the arguments are for our default A-sign transformation, once again, we can check using the little question mark. And we can see right off the bat it's returning a kind of wrapper function, so not actually providing the arguments that we need. However, we know that wrappers will often be pointing at a similarly named function, so with a little bit of investigation work, we can find that leaving off the underscore trans, there's a different function, flojo which is actually provides us the individual arguments that we use for mass cytometry data. So m corresponding to the numeric full width of the transform display, t being the numeric maximum value, a being the numeric additional negative range, and length being maximum value of the transform data. And then you can see the defaults are being specified within usage. So kind of similar to what we did for SFC, we can transfer in those defaults so that we're better able to adjust them. As we currently have the transformation applied, this is what we're getting for the visualization of our CD4 by our CD8 population. And we can see that sure enough, this is what's actually being applied in the background as far as the math formula that's happening every time the transformation is happening. So if we go ahead and check right now, remember m is basically asymptotic decades. It's currently set to 4. We're just going ahead and tinkering back and forth to see what happens. So if we increase this to 5, you can basically end up seeing we're actually separating out the positive CD8 and CD4 from the rest of the background values that are basically set around 0. So we're getting a little resolution here and there. Vice versa, if we decrease it down to 3, it gets a little bit shrunken back towards the axis, a little bit in that range as well. So t basically being, I think that was the higher value if I recall correctly. So if we increase it to 24,000, we see this is what we currently look like. If we end up decreasing it, it's shifted a little bit over here, but not quite as much. This is where those who are doing with mass cytometry data everywhere can be able to pinpoint more of the actual changes that are needed. Negative data, most of the values for mass cytometry data will be around 0 for most of them. We can add negative decades, but as you can see, the plot doesn't really change dramatically for most of these. There's a little bit of change, but not too much. And versus if we end up setting it at 0, this is the plot that we end up seeing. And length, the max value, let's double it to 512. You can see it's changing a little bit as far as the actual numbers being displayed on the scale, but the shape's not changing too dramatically. Versus if we end up reducing it to 128, yeah. You can see basically these arguments will allow us to fine tune based on whatever situation you have. In this case, this data seems to be working pretty well for these particular markers, so less adjustments needed to be done. So far we've just looked at the transformations in context of visualizing our data, and this is very much an important aspect because that then allows us to be confident in the gates that we placed and be able to denote a positive from a negative population. However, one other area that we're going to encounter transformations is a couple weeks from now in the course when we're actually extracting out fluorescent signatures from our single color controls to use for unmixing. And that can matter whether you're accidentally passing in transformed data versus just the raw parameters. So the way you actually go about extracting data from a gating set is we need to take it back a step. So instead of working with a gating set object, we need to return it back to what's known as a cytoset. And the way you do that right now with the flow workspace, kind of like what we saw a couple weeks ago, is the gs underscore pop underscore get underscore data function, basically passing it the very first specimen in our cytoset, subsetting it down, and using root to grab all the cells that are present. This now becomes a cytoset object that we've encountered in the past. And then we can use the express function that we've played around with in both week 3 and week 5. And you can see we're actually being able to get back the underlying data set accordingly. Now, if we're looking at these values after a little bit, especially if you have a lot of practice looking at flow cytometry data, you can see these all are kind of pretty uniform. They're all roughly around 2,000, 3,000 or so in value, which is not very much when it comes to like a bioexponential scale. So right off the bat, you can start seeing this is kind of like the transformed data. It's not actually showing you the untransformed data that can be logs of the difference between individual cells. So unlike commercial software, where often the transform is applied separately, it's applied on top, so just changing how we visualize the data. In the context of transformations that happen within R, it is actually going and transforming the values that are present within the column, which is why that inverse transform is very important because it allows us to go back to what the original values were before we actually went in and transformed the values present. So when it comes to a couple weeks from now, when we're actually working with data, the main thing we need to do is we need to make sure when we are extracting out the raw data, we need to untransform it back to the original state, in which case we'll be using this inverse.transform argument. So if we look at how this will look like in a couple weeks, basically to that function, we're adding on this additional argument, inverse.transform is equal to true, which will revert it back to the original shape. So then if we're actually looking at the data, it's going to look much more like how we had it originally. This is not something we need to worry about right now, but just putting it on your radar as the status that your transform data is currently in will matter when you're working with it in R. All right, so our takeaways for this particular week is we looked at how to set up the underlying data for transformation within the Flow workspace context by providing both the transform that we're interested in applying as well as the column names that we're interested in transforming, creating a transformer list that then gets applied to our gating set. We also investigated how to visualize our data as well as customize the arguments just to make sure we're optimally transforming the data rather than just willy-nilly accepting the defaults, which can end up with weird kind of visualizations. And we also did venture into how to update our metadata, and in the process had a brief exposure to what a regular expression is when it comes to extracting out value. So in terms of how to go about best applying transformations, oftentimes the best approach is just to know for your particular instrument, what is their range settings, what are the typical parameters, and then apply those when you're first setting up your R experiment. Vice versa, if you're working with data that's not your own, that came from like a repository somewhere, you just need to know what are the main important ones, especially like width, and then be able to adjust the others so that your visualization makes sense in context of what you're trying to do. So proper transformation in the context of a supervised analysis where you're doing manual gating will often be very different from if you're relying on a more of an unsupervised algorithmic style analysis where the actual transformation may affect the scale, which may affect some of the dimensionality reduction approaches. So next week, we don't have a class. I will be presenting at the ABRF conference. So if you are attending ABRF, please drop by. I'd be happy to talk to you. This week, I had originally intended to look at both transformation and compensation, but as you can see, we're at the top of the hour only discussing transformation, so the compensation lesson will have to wait. I haven't yet decided if I'm going to try to do it as a one-off course, because it's mostly focused at those who do conventional flow cytometry, or if I'm going to try to put it into the sequence before we end up getting to the manual and automated gating. So I'll think about that over the next couple weeks, and I'll let you know either way. We will still cover how to do compensation, but I just don't know if it's going to be in the context of our 30-week schedule or it's going to be a one-off that for those who are interested can go and check out. As a heads up, next week I'll be sending out the first class feedback survey. So this is our very first year running the Cytometry in R course, and any feedback that you can provide us will make us help address any areas where we're falling short, things that need to be improved on, as well as once this course has concluded and the resources are available for those who may be self-studying in the future, how to make those particular resources better when there's no active person to be able to go and check with. And that should be sent out sometime next week. And with that, we're at the top of the hour. I'm conscientious of everyone's time, so if you need to go, please feel free to head out. The additional resources section, the focus of today was very much more of how to go about applying the transformation in R, because that's the section that can be a little convoluted. Now, what is the best transformation? What's the best arguments for your intended purpose? That's something we can't really address in a general manner, but fortunately, this is an area where a lot of the existing resources are already present and are quite good places to start. So for example, if you look at either the FlowJo or the FCS Express transformation manuals, you'll see a good explanation for the different methods as far as what's done. Optimizing transformations for automated high throughput analysis for cytometry data was quite useful, so I've linked it there. The Spectra R package, run by Thomas, Felix, and Giovanna, has really lots of different transformations that are applied within the context of their pipeline. So their help vignette documentation for data transformation is really good, especially with the visuals. So I went ahead and linked it there. Same thing for Oliver Burton's Colibri Cytometry blog. He has a section for scaling your data for dimensionality reduction, which can be useful just to kind of see like what happens when you're not properly scaling a data set. As I mentioned at the beginning, Felix came out for the Cytobytes channel with a transforming cytometry data video. I think it's only 6 minutes. It's worth your watch. And for those of you who primarily work with mass cytometry and CyTOF data, this link is a goldmine. It links to a bunch of other R workflow as far as the actual process, so I recommend it if you're trying to figure out how to best apply your R-sign transformation. The optional take-home problems for this week. So the very first one is we have worked with the spectral flow cytometry transform, but as you saw, we didn't include forward scatter, side scatter, or time. This can be something that oftentimes you'll do inadvertently, especially if you didn't like subset down the column names. So go ahead and accidentally apply a transformation to these parameters and visualize them just to see what happens when they accidentally end up transformed. For problem 2, we had time to look at the bi-exponential transformation, but the other one that's commonly used, especially in R, is the logical transformation. So kind of repeat the process we carried out for bi-exponential using the logical just to see how that affects things. Take screenshots and compare the difference. And for problem 3, there's 2 assigned default transformations within Flow Workspace. They look a little bit different in terms of their arguments, so just kind of same thing, play around with it, apply it to your individual panel just to see what the differences are as far as what you see. And with that, that's all I have for today. If you have any questions, I'll be in the chat for a little bit. If not, please head to the discussion forum. If not, I'll see you in 2 weeks for either compensation or automated and manual gating, whichever I decide to do for the class in 2 weeks.